Oh, hello. I didn't see you there. I was just reading this fascinating article about how pH affects mitochondrial dysfunction. Would you like to hear about it? No? Well, too bad. My friends and I are going to tell you about it anyway. Two organelles are linked to cellular aging and to each other, the mitochondria and the vacuole. As we know from class, the mitochondria is the energy factory of the cell and produces most of the cell's ATP. It has an inner membrane where ATP synthesis occurs due to a proton gradient between it and the outer membrane. This is called the membrane potential and is very necessary for proper mitochondrial function. The vacuole is a storage organelle that can store a variety of things, including amino acids, ions, and other molecules. In this study, cell age is defined as the number of times a cell is divided. It is well known that a symptom of cell aging is increasing mitochondrial dysfunction. As this study will go on to show, the acidity of vacuoles will have an effect on how quickly mitochondria become dysfunctional, at least in the yeast cells that this study focuses on.
The VATPase pump uses ATP to transport H+ molecules, or protons, from the cytosol into the vacuole. The higher the concentration of protons inside the vacuole, the higher the vacuolar acidity. This experiment investigated what genetic and biological factors delay the onset of mitochondrial dysfunction by aging. To begin, 250 genes in the budding yeast S. cerevisiae were overexpressed in a screening test to understand which gene functions have a marked effect on this yeast longevity. Through this screening, two genes were identified to increase lifetime and decrease mitochondrial dysfunction, VMA1 and VPH2. These genes are known to affect the function of vacuolar H+ ATPase pumps, or VATPase. When VMA1 was overexpressed, age-induced mitochondrial dysfunction was suppressed in 93% of aged cells. The same was true for 77% of cells with the overexpressed VPH2 gene. This was then linked to the vacuolar acidity of the cells, as the overexpression of VMA1 and VPH2 prevented decline in vacuolar acidity in over 70% of aged mother cells. How VMA contributed to this is unknown, but VPH2 caused more VATPase to be assembled at the membrane of the vacuole. Deficient vacuolar acidity in mother cells can be passed to daughter cells, causing a lack of VATPase and a lower overall membrane potential. This is the part of the study that demonstrated the close link between vacuolar and mitochondrial function. Vacuolar acidity is known to be necessary for efficient protein degradation and for ion and amino acid storage. Protein degradation was blocked independently of vacuolar acidity, and this was shown to have no visual effect on mitochondrial longevity. Next, the transporter proteins of neutral amino acids were inhibited in the cells, independently of vacuolar acidity, by overexpressing and suppressing the ABT1 gene. This was shown to have a marked effect on mitochondrial longevity. The overexpression of ABT1 kept the mitochondrial structure and membrane potential of aged cells at a constant in 63% of the cells tested. The deletion of this gene caused mitochondrial dysfunction to develop at an accelerated rate, independent of vacuolar acidity decline. It was also found that ABT1 transports amino acids through the VATPase pump. Finally, the yeast cell was subjected to calorie restriction to observe if this has any effect on vacuolar acidity and mitochondrial longevity. Calorie restriction worked to suppress decline in vacuolar acidity, which increases the longevity of the mitochondria and subsequently the cell itself. Calorie restriction was also prevented in a strain of cells lacking VATPase activity and did not further change the longevity of cells with overexpressed VMA1. Each of these results suggests that the vacuolar acidity is directly related to mitochondrial function and the longevity of cells.
Of the 250 genes which control structure of the mitochondria, the genes VMA1 and VPH2 delayed structure degradation when overexpressed. These two genes specifically affect the function of the vacuolar VATPase pumps, which regulate vacuolar acidity. As cells age and begin to show dysfunction, vacuolar pH increases or becomes less acidic. Overexpression of VMA1 and VPH2 prevented this rise in pH, which in turn slowed mitochondrial dysfunction. pH affects mitochondrial function because it triggers VATPase pumps, which transport ions and amino acids into the vacuole. When the proton gradient, or pH difference, across the pump was reduced and treated with overexpression of the amino acid transporter AVT1, mitochondrial dysfunction in 60% of cells was prevented. The increase in pH as a cell ages prevents AVT1 from importing adequate amounts of amino acids across the VATPase pump. Overexpression of AVT1 prevented alterations in mitochondrial structure, and without AVT1, overexpression of VMA1 and VPH2 had no effect on the rate of mitochondrial dysfunction. Caloric restriction prevented the increase in vacuolar pH by conserving nutrient-sensing pathways. Overall, the study found that age-induced mitochondrial dysfunction is not the result of a block in vacuolar protein degradation, but is due to decreased uptake of amino acids into the vacuole, which is in part caused by an increase in pH.